So think of your brain as like the ultimate VIP club.
Oh, I like that analogy.
Right. It's guarded by the most intense, uncompromising security system on Earth, the blood-brain barrier.
Right, the bouncers at the door.
Exactly. And for decades, we've thought of this barrier as just an impenetrable concrete wall. The rule was simple, you're either inside the club or you're out.
Yeah, that was the textbook standard.
But what if we told you there are secret tunnels? Like hidden back doors and skylights that specific immune cells use to basically sneak in and out right under the bouncers' noses.
It completely upends our classical understanding of brain anatomy. I mean, truly. We're finding out that the fortress isn't totally sealed,
Right.
and the implications for human health, aging, and disease are just staggering.
Welcome to today's Deep Dive. I'm your host, and I'm here with our resident neuroimmunology expert.
Hey there, so glad to be here for this one.
Oh, me too.
Yeah.
So if you are joining us today, we are looking at a fascinating stack of research. It centers on this dense, cutting-edge scientific review titled Gateways for Myeloid Cell Entry into the Central Nervous System by Lucas Amann and Marco Prince.
A fantastic paper.
It really is. And our mission for this deep dive is to take this incredible, complex text and map out exactly how your brain's immune system is populated. We're going to trace this from before you were even born, all the way to how these secret gateways change as you age.
And, we aren't just doing a biology lesson for the sake of it here.
Right, there are huge stakes.
Absolutely. Understanding these hidden pathways is, it's the absolute key to curing neurodegenerative diseases and brain inflammation. If we want to fix a sick brain, we have to know exactly how immune cells get in.
And what rules they follow once they cross the threshold, right?
Right, exactly.
Well, I am so ready to this. But I feel like to understand the intruders who might sneak into our VIP club, it makes sense that we first have to meet the resident guards.
That's a great place to start.
So let's talk about the brain's native defenders. The paper focuses really heavily on myeloid cells.
Right. So myeloid cells, specifically macrophages, are your body's first line of innate defense. They eat cellular debris, they fight off pathogens, that sort of thing.
Okay.
Now, to understand the brain's immune system, you really have to visualize a fundamental geographical divide.
Geographic divide.
Yeah, imagine the brain in two parts. First, we have the parenchyma.
The parenchyma.
Right. That is the functional, delicate tissue of the brain where the neurons and astrocytes live and do their computing. That's, the inner VIP room in your analogy.
Okay, the actual dance floor.
Exactly. And in that highly exclusive room, there is only one type of immune cell allowed. They're called microglia.
Microglia, so they are the elite inner circle guards.
Spot on. They are the only ones allowed on the dance floor. But outside the parenchyma, at the very borders of the brain, we have a completely different group.
Okay, the perimeter security.
Yeah, these are called CNS-associated macrophages, or CAMs. CAMs. Okay, so if the parenchyma is the VIP dance floor, the CAMs are the security guards working the velvet ropes, the perimeter walls, the parking lot.
You got it.
Because the review actually maps out 4 distinct niches where these border guards live, and it's pretty fascinating to picture. For instance, there are leptomeningeal macrophages.
Yes, so those reside in the subarachnoid space, which is basically this fluid-filled cushion wrapped entirely around the brain.
Okay.
Then you have the perivascular macrophages. If you think of the blood vessels penetrating the deep brain tissue as plumbing pipes,
Right.
these macrophages are stationed in the tiny spaces right along the outside of those pipes, just constantly patrolling.
Okay, so that's the fluid cushion and the pipe perimeters. Then the authors mention epiplexus and stromal macrophages.
Right. Those are located inside the brain's ventricular system and the choroid plexus.
Which is exactly.
You can think of those areas as the internal water tanks where your cerebrospinal fluid is actually produced.
Oh, wow. Okay. And finally, there are the dural macrophages. They live in the dura mater, which is that really tough, leathery outermost membrane right underneath your skull bone.
Okay, let me push back on this for a second, because reading through this, it almost felt like an avalanche of terminology.
Need a lot of names.
Right. If they're all basically just macrophages sitting at different borders of the same organ, why do scientists insist on categorizing them with a half dozen different names? Like, does it actually matter?
It's a completely fair question. It feels like splitting hairs, but the distinction is actually crucial.
Really?
Yeah, location entirely dictates biology here. What's fascinating is that these cells express completely different molecular markers, and they play by different rules depending on where they are stationed.
How so?
Well, for instance, the CAMs, working the borders, express a specific identification marker on their surface called the macrophage mannose receptor.
Or CD206, right?
Right, exactly, CD206. You can think of it like a specific security badge, but the microglia, the guards inside the inner parenchyma, they don't have that badge.
Oh, interesting.
But even more importantly, they follow completely different rules for how they are replaced when they die.
Tell me more about that. What do you mean by replacement rules?
Well, your body is constantly replacing old cells, right?
Sure.
But the microglia in the inner VIP room, along with the leptomeningeal and perivascular macrophages, basically never replace themselves with outside cells from your circulating blood.
Wait, never?
Basically never. They are totally self-sustaining. When one dies, another one native to the brain splits to replace it.
Oh, wow.
But the macrophages out in the dura mater and the choroid plexus, they operate completely differently. They are constantly accepting new recruit-specific immune cells called monocytes straight from the bloodstream throughout your entire life.
Okay, let me just make sure I'm wrapping my head around this for a quick second.
Sure.
So to recap for you listening, essentially, we have the microglia guarding the inner sanctum and these various CAMs guarding the borders. But they play by entirely different rules depending on their exact zip code.
Right.
Some of them just self-replicate forever, keeping the doors locked to outsiders, while others are actively pulling in new recruits from the blood.
Spot on. The rules change depending on where you stand in the fortress.
But that creates a massive logical puzzle, doesn't it?
How do you mean?
Well, if the inner sanctum is completely closed off to the blood and microglia just maintain their own population in total isolation, how do they get in there in the first place?
That is the big question. And to answer that, we have to travel way back in time, like to before you were even a fully formed fetus. We're talking about very early embryonic development.
Okay, because the central dogma I remember from high school biology is that all of our immune cells come from our bone marrow.
That is the classical textbook answer, yes.
But not here.
Not here. The brain's core immune cells, the microglia, and most of those self-sustaining border CAMs we just talked about, they do not come from your bones. They originate from an extra embryonic structure called the yolk sac.
The yolk sac? I mean, it sounds almost prehistoric.
It is a very ancient lineage. And the timeline here is incredible. At about embryonic day 8.5.
Which is very, very early in development.
Extremely early. So at E8.5, a specific type of stem cell arises in the yolk sac. And over the next 24 hours, these cells go through this rapid, dramatic maturation process to prepare for a massive journey.
Yeah, the paper spends a lot of time on this specific maturation phase, calling it a shift from an immature stage to a mature stage. What is actually happening to the cell during that transition to make it ready to move?
Think of it as the cells changing their molecular uniforms. When they first appear, they are wearing a stem cell badge, a protein receptor that tells them to basically stay put and multiply.
Okay.
But as they mature, they literally turn off the gene for that stem cell badge. At the exact same time, they start manufacturing mature immune badges and navigation sensors on their surface.
Oh, so they're gearing up for the journey.
Precisely. By embryonic day 9.5, they are fully prepped pre-macrophages ready to migrate out of the yolk sac.
So they pack their bags, change their uniforms, and migrate to the developing brain. But here is the part that absolutely blew my mind when I was reading this.
The blood vessels.
Yes. At embryonic day 9.5, the blood-brain barrier isn't built yet. The vascular roads, the blood vessels, aren't even fully paved. It's like a construction crew moving onto a site before the highways are even laid down. How do they physically get into the brain tissue without blood vessels to carry them?
It's a great analogy. Because the blood vessels aren't fully formed, these pre-macrophages don't just float in on a bloodstream highway like normal immune cells do later in life.
Right.
They have to literally crawl through the surrounding embryonic tissue.
Crawl.
Yeah, to get into the brain, they physically breach the delicate inner membranes or squeeze through the primitive lining right into the developing ventricles.
And they don't just politely squeeze through. I read they actually use specific enzymes called matrix metalloproteinases, or MMPs.
Yes. Think of those MMPs as molecular machetes.
Molecular machetes. That's a great image.
Right. The cells actively secrete these enzymes to dissolve the extracellular tissue in front of them, clearing a physical path into the developing brain. It is an active invasion. And here's the beautiful part.
What's that?
Once they hack their way into the inner sanctum, the local environment of the brain tissue starts giving them chemical instructions.
Meaning the tissue tells the cell what its final identity should be.
Exactly. If the cell settles in the inner parenchyma, the local environment tells it to completely drop that border guard badge.
The CD206 marker.
Exactly. Drop it entirely and become a true microglia. But if it settles on the border, it keeps the badge. The local niche drives their ultimate identity.
That is just incredible. Okay, so to synthesize this for a second, your brain's primary immune cells, microglia and most of those border guards, are an ancient lineage born in your embryonic yolk sac. They literally crawled into your developing brain using molecular machetes before your blood vessels were even finished. They are entirely separate from the immune cells being made in your bone marrow today.
Which brings up an unavoidable paradox.
Right, because the fortress is now built.
Right.
The VIP room is populated. Fast forward to adulthood, and the doors are securely locked by the blood-brain barrier. So how are we seeing new cells interact with the brain later in life? If the club is perfectly sealed, what happens when new immune cells need to get in?
And this is where recent discoveries have completely shattered the old textbooks.
I love when that happens.
Me too. While the blood-brain barrier does an excellent job of blocking the main vascular roads, scientists have just mapped out completely new alternative back channels that bypass the main barrier entirely.
I have to admit, reading the section on these alternative routes, the one that made me physically drop the paper on my desk was the discovery about the skull bone channels.
That's wild, isn't it?
It is.
Yeah.
We always think of the human skull as just a solid, dumb helmet.
Yeah.
But the authors are saying it's porous.
It is actively connected. We used to think the skull bone and the brain were completely separate compartments, but it turns out the bone marrow inside your cranial skull and your vertebrae has specialized, direct, vascularized channels.
Like tiny anatomical tunnels.
Exactly. Tunnels that go straight through the solid bone to the brain's outer surface, the dura mater. Neutrophils, B cells, and monocytes use these microscopic bone tunnels as a fast track to jump directly from the skull marrow to the brain's borders, bypassing the systemic bloodstream entirely.
A literal tunnel through the skull bone. That is wild. But that's just the first one. They also discuss something called ACE points.
Right, arachnoid cuff exit points. This one is highly mechanical.
How so?
Inside the brain, you have these bridging veins that drain venous blood away from the brain tissue and out into the big dural sinuses. Where these veins breach the arachnoid membrane, there is a physical discontinuity in the barrier layer. It's not perfectly sealed.
Okay.
Think of it like a loose cuff on a shirt sleeve where the hand pokes through.
Hence the name, arachnoid cuff exit points.
Right, and high-resolution imaging suggests immune cells might use these exact ACE points. They slip out of the dura mater, squeeze through that loose cuff, and drop right into the subarachnoid space.
Wow. And then there's a third major staging area they highlighted deep inside the brain called the velum interpositum, or the SIX.
The SIX is fascinating. It's a membranous structure formed by a folding of the pia mater deep in the brain's anatomy.
And what happens there?
Well, studies are showing that during times of neuroinflammation, monocytes accumulate heavily in this folded area. It seems to act like a hidden staging ground where immune cells gather, and from there they might jump directly into the surrounding brain parenchyma.
Okay, I have to stop and ask the obvious question here, because from a biological standpoint, this sounds like a massive security flaw.
It does.
Why would evolution leave these back doors open? Is this strictly designed for emergency defense, like a fire escape for your immune system? Or are things like pathogens and diseases using these tunnels too?
That is the dark side of this anatomical discovery. It is absolutely a security flaw. Bacteria that cause meningitis, which are blood-borne, have been documented accumulating at the dural sinuses and hijacking these exact routes, like the ACE points, to invade the brain.
So they use the loose cuffs to slip past the barrier.
Exactly. Even leukemia cells have been shown migrating from the cranial bone marrow, crawling down those exact skull channels to infiltrate the brain's wrappings.
Oh, wow.
So yes, pathogens and cancers are actively using our own secret tunnels against us.
That is terrifying. So we have this adult brain that we thought was a perfectly sealed vault.
Yeah.
But really, it has hidden highways connecting the solid skull bone directly to the brain's wrapping. It has loose cuffs around its veins and folded membranes acting as staging grounds.
Right.
And basically, immune cells, and unfortunately pathogens, can bypass the blood-brain barrier entirely.
Which sets the stage perfectly for what happens to these gateways over time. because, a security system is only as good as its maintenance.
Right. If these tunnels and barriers exist, what happens as we get older? We all know biological systems start to rust.
Well, if we connect this to the bigger picture of aging, physiological aging physically changes the architecture of the brain's borders. For instance, the ependymal cells that line those internal water tanks, the ventricles, they start to lose their tight junctions. The molecular glue holding those cells tightly together simply degrades over time. Furthermore, the blood-brain barrier itself slowly becomes leaky and less efficient.
And the review highlights a fascinating study about exactly this concept, using aging mice. And they use a really specific genetically engineered mouse model called the MS4A3 mouse.
The MS4A3, yeah.
What is the logic behind using that specific mouse?
It comes down to tracking. If you want to know if cells from the blood are entering the brain, you have to be able to tell them apart from the cells that were born in the brain.
Right, that makes sense.
What the MS4A3 genetic tool does is permanently attach a fluorescent marker to monocytes derived from the adult blood. It acts like a permanent GPS tracker. Every immune cell born in the adult bone marrow glows.
Okay, so they have a glowing tracker on the blood cells. What did they find when they looked at the brains of these mice as they got old?
Well, in healthy young mice, you don't see those glowing blood cells in the deep brain tissue. The fortress holds. But as these specific mice aged, those glowing blood-derived cells started showing up inside the inner VIP room. They were engrafting into the brain tissue as microglia and CAMs.
So the fortress was actively leaking.
Yes. Natural aging alone allowed outsiders to bypass the bouncers and take up residence in the parenchyma.
That makes sense for natural aging.
Yeah.
But how does this apply to severe human diseases? The big one that comes to mind is Alzheimer's disease. When amyloid plaques build up in the brain, do these blood cells flood in through the secret tunnels to help clear the plaques, or do they make the inflammation worse?
That question is at the center of one of the most intensely debated controversies in neuroimmunology today. It is a massive, unresolved civil war in the scientific community.
Really?
Oh, yeah. Different labs are getting entirely contradictory results, and it all depends on which genetic mouse model they use to track the cells.
Walk us through this fight. Why is it so hard to get a straight answer?
Because fate mapping, which is that process of putting a permanent genetic tracker on a cell, is incredibly tricky. On one side of the debate, you have studies using specific genetic tags called CX3CR1 and CCR2.
Okay, those are the tags.
Right, and they crossbreed these tracking mice with Alzheimer's mice. These studies show absolutely no peripheral blood cells entering the brain parenchyma. The researchers argue that the resident microglia, the native guards, handle the Alzheimer's plaques entirely on their own, and the blood cells stay out.
Okay, so that side says the fortress stays sealed even during Alzheimer's, but what about the other side?
The other side uses a different genetic tag called LFLT3 fate mapping, and they see the exact opposite.
No way.
Yeah, their tracking shows blood-derived cells actively pouring into the brain and clustering around the amyloid plaques, physically altering them. So the scientific community is actively fighting over a fundamental question. Are invading blood cells trying to help? Are they causing damage? Or are they even getting in at all?
And the slight difference in how a genetic tag behaves alters the entire conclusion. That is wild to think about.
It really is.
But, the paper doesn't just stick to mouse models. It also talks about human pathology, specifically a concept called clonal hematopoiesis. What exactly is that?
Clonal hematopoiesis is a phenomenon that happens naturally as humans age. Mutations begin to occur in the blood stem cells located in your bone marrow.
Okay.
These mutated stem cells create mutant clones of immune cells that then circulate freely in your bloodstream. What's chilling here is that researchers have examined the postmortem brains of humans who died with Alzheimer's disease and Lewy body dementia, and they found these exact mutant blood cells deep inside the brain tissue.
Wait. Mutant blood cells from the aging body are being found deep inside the inner sanctum of human Alzheimer's brains.
Yes. And the connection between circulating blood mutations and brain disease gets even more direct.
How so?
There is a rare human disease called histiocytosis. Patients with this condition have a very specific mutation called BRFV600E in their circulating myeloid cells. This mutation causes those circulating cells to actively break down the blood-brain barrier. They essentially kick the doors in, allowing a flood of mutant cells into the brain, which causes severe, late-onset neurodegeneration.
So in that case, the intruders are literally breaking down the doors to the club.
Exactly.
Okay, so zooming out for a quick recap here. Aging and disease blur the lines between the body's peripheral immune system and the brain's private guards. The barrier gets leaky, the loose cuffs get looser, and mutant blood cells can sneak in. But scientists are still trying to definitively prove whether these invading cells in conditions like Alzheimer's are the heroes or the villains.
Exactly. Which brings us to the ultimate so what of this entire review. Why does all this incredibly deep anatomy matter to you, the listener, right now?
Right. Why do we care about yolk sacs, ACE points, and skull channels?
It matters fundamentally because of how we design therapeutics and drugs. Take multiple sclerosis, an autoimmune disease where a patient's own immune cells attack their brain. We have a highly effective drug called natalizumab. It works by blocking a specific integrin on the surface of T cells. It essentially changes the locks on the blood-brain barrier to stop those specific T cells from entering the brain and causing damage.
So it changes the locks on the front door. But the paper points out that natalizumab completely fails to block myeloid cells.
Precisely. It doesn't stop macrophages or monocytes. Why? Because as we've learned today, myeloid cells don't use the front door.
Ah, they use the tunnels.
Right. They use different biological rules, they use different molecular machetes, and they use totally different secret tunnels to get in. If you want to stop harmful myeloid cells from entering the brain during a disease, you can't just lock the main vascular highway. You have to know which secret tunnel they're using and block that.
And on the flip side of that coin, what if we want to get cells into the brain? You mentioned earlier that the holy grail of this field is curing genetic brain diseases.
Right. Think about replacement therapy. If a patient is born with a genetic disease where their native microglia are defective and failing, the logical cure is to replace those broken guards with healthy, engineered cells.
Sure, that makes sense.
But right now, the only way we know how to do that is incredibly brutal. It requires a patient to undergo a whole-body bone marrow transplant combined with incredibly harsh radiation or chemotherapy conditioning.
Oh, wow.
Yeah, you essentially have to nuke the body's entire immune system just to force the new healthy cells across the tight blood-brain barrier. It is highly toxic and dangerous.
Oh, hold on. This is the part where the puzzle pieces snap together.
Yeah.
If we now know about the velum interpositum,
Yeah.
if we know about the loose arachnoid cuffs, and most importantly, the physical vascular channels in the skull bone, we don't have to nuke the whole body. Can't we use those exact secret tunnels to smuggle the cure right past the bouncers?
That is the exact aha application that makes this research so groundbreaking. That is the cutting-edge frontier of neuroimmunology.
That is amazing.
If we can map out and understand the exact molecular signals that open those skull bone channels, we could theoretically inject a therapeutic, genetically corrected macrophage directly into the cranial bone marrow.
And just let it walk through the natural secret tunnel into the brain.
Exactly. Completely bypassing the systemic bloodstream and completely eliminating the need for whole-body chemotherapy.
We literally use the fortress's own security flaws to deliver the cure. That is absolutely brilliant.
It really is.
Yeah.
A precise understanding of these gateways isn't just obscure anatomy trivia. It is the literal blueprint for the next generation of targeted cell therapies for the brain.
Well, we have covered an immense amount of ground today, from the macro down to the microscopic. Let me just try to synthesize this incredible journey for everyone listening.
Go for it.
We started way back at embryonic day 8.5, watching an ancient lineage of immune cells change their uniforms, crawl out of the yolk sac, and basically use molecular machetes to build the brain the very first security force before the blood vessels even existed.
Right.
We mapped the exclusive VIP room of the microglia and the complex perimeter guarded by the cams. We discovered that the adult brain is not a static, perfectly sealed concrete bunker. It is actively dynamic, riddled with secret skull tunnels, leaky arachnoid cuffs, and folded membranes.
Yeah.
And finally, we saw how the rusting of this security system during aging and disease might just be the key to designing revolutionary non-toxic cures.
The brain is constantly communicating with the bone marrow right next door, and we are just now finally finding the maps to these hidden highways.
It completely reframes how you view your own biology. But before we sign off, I want to leave you with one final lingering question to ponder based on the incredible sources we explored today. We learned that the bone marrow inside your solid skull has direct physical tunnels straight to your brain's delicate immune system, constantly feeding it cells and signals.
Right.
Which makes you wonder, does a simple physical bump on the head or the overall biological health of your skull bone itself silently dictate the long-term health of your mind?
It really changes how you think about the relationship between your bones and your thoughts.
Definitely something to think about the next time you put on a bicycle helmet. Thank you so much for joining us on this deep dive. Until next time, keep questioning and keep exploring.